Dear students, this is Dr. Pooja Thakkar, a biology expert, and we are in the 7th session of cell biology, that's on cytoskeleton. Now, what do you mean by this particular word? Cyto is related to cell, and skeleton is giving stiffness or the basic building framework. Okay, so the framework or the structure which is giving a particular kind of shape as well as the structure to the cell is called cytoskeleton, that is making the cell's integrity. Okay, now cytoskeleton is basically a fibrous fine tubular structure which form the supportive structure as we have discussed to the cell. There are basically three kinds of the fibers. They are basically proteinaceous in nature. They are of three types. The first one is called microtubules. Here you will be able to notice green colored structure which are labeled as a polar microtubule or the astral microtubule, which are originally arising from a particular center called MTOC, that is microtubule organizing center. Okay, so these centers are basically located in one organelle, namely centrosome. They are present in each of the cell, and during the time of cell division this particular center is activated to form microtubules, which are proteinaceous structures. So microtubules are unbranched hollow tubules which are of indefinite length. That means they can be polymerized to make a longer chain. They can be depolymerized to make it very shorter. As per the need, they'll form. They are basically of 25 nanometer thickness, as well as they are having the central core of 15 nanometer. Now the boundary, which is formed by 13 helically arranged protofilaments, which are made up of alpha and beta tubulin. This boundary is basically, you will be able to notice in this particular diagram that these tubules are basically hollow. The outermost boundary is made up of 13 protofilaments. Now these 13 filaments are made up of two kinds of monomers, that is alpha tubulin, which is green in color, and the beta tubulin, which is yellow in color. They orient themselves in turn by turn manner to form the overall peripheral located bulbs, and into which you will be having the central empty core. These kinds of tubules can quickly grow and get longer, or they can get shortened very quickly as per the need, as I've discussed. So they are mainly involved in dragging the chromosome to the respective poles, or else they are pulling the chromatid molecules so that the particular chromosome has been broken up by the centromere and have the two chromatids which are splitted, and they'll travel towards the opposite poles during the process of cell division, so that each and every cell can get the exact quantity of the genetic material. Now how the process is happening? The main process is happen, that is the assembly process or the polymerization process is happen in the presence of calcium, magnesium and GTP, that is guanosine triphosphate. So they are aiding the assembly to grow more and more filaments, but colchicine is one of the molecule that will prevent the formation of microtubule, so they are aiding in shortening of the filament. Or you can also say that colchicine is the major chemical which is pausing the microtubule formation at a particular time to make the chromosome as it is when the particular kinds of chromosomes are located in the central plate called metaphase plate, so that you will be able to study each and every chromosome in very detail. Now these kinds of microtubules are served as the basic structure. So these are the very basic and common structure found in centrioles. As we have discussed, they are forming microtubules for the cell division process. The second important thing is they are also found in cilia. They are also found in flagella, as well as they are forming spindle apparatus. Now what do you mean by spindle apparatus? Spindle fiber formation, that is microtubule formation. Both the things are same, which are mainly involved in cell division process for the segregation of chromosomes. So these are the microtubules which are providing specific shape as well as polarity to the cell. They're helping in cell movements as they are dragging their cells into particular orientation so that the cytoplasm will get stretched simultaneously and the cell can move in particular direction. They're also aiding in chromosomal movement, as well as they are helping the intracellular material to transport from one place to another place. And this is the second diagram where you will be able to notice the polymerization, in which the alpha and beta tubulins are added and they are removed during the process of depolymerization. So these are the process they are happened into the cell as per the requirement. Now the other kind of cytoskeleton is called microfilaments. You will be able to notice these microfilaments in red colored structure. They are looking like a DNA double stranded structure coiled on each other. So these are the double helical cylindrical rods, occurs typically in the muscular as well as the non-muscular cells. They are of indefinite length as the microtubules, and they are having the diameter of 8 nanometers. They are mainly help in cytoplasmic streaming. Now what do you mean by this? Cytoplasm is get stretched at particular orientation so that the cell can move in that particular direction. The other movement is amoeboid movement, for example pseudopodia formation by the cell, that has happened by the help of these microfilaments. The sol gel changes, that is the solution to gel changes or gel to solution changes. The cytoplasm of the particular cell is basically a fluid material. They can also be in the jelly version, so as per the need they can be very liquidy or very much in the gel version, gel based material. So that kind of sol gel changes can happen by the presence of microfilaments. Gliding of cell, that is the same thing as the cytoplasmic streaming or the amoeboid movement, you can say. They are helping the cell to glide from one place to another surface. And the last function of these microfilaments is cleavage, that is the cell division. So during the cell division process they help the cell to segregate into two cells. In the diagram also you will be able to notice they are made up of a small monomer called F-actin. See, basically these kind of microfilaments are basically of two types, that is actin and myosin. Basically actin's diagram is given over here. The monomer of this particular kind of filamentous actin, that is F-actin, is the polymer. Now that is made up of a small monomer unit called globular actin, which is smaller and you can see it in the round shape. So these kinds of rounds are attached to form the polymer like structure by the utilization of ATP as the energy. Now the last category of the cytoskeleton is the intermediate filament. These are the solid as well as unbranched protein fibrils, and they are of 8 to 10 nanometer in strength. They are of basically four types, that is keratin, that we already know, that is present in our hair as well as nail. The neurofibrils, the glial filaments, and the heterogeneous filaments. Okay, so basically you will be able to notice that these filaments are for example keratin intermediate filamentous network has been seen in the animal cell, and these kind of animal cells micrograph is easily able to show you the keratin filament and their orientation. So varieties of cells are having different kinds of intermediate filaments, like epithelial cells are having keratin, astrocytes are having glial filaments, nerve cells are typically having neurofilaments. In certain kind of cells vimentin kind of filament is present, and in few muscle fibers desmin is present. Okay, so these are the variety of intermediate filaments. Based upon the type of cell and their function, they'll be given different names. So in general, if you want to conclude the thing, you will be able to notice that the microtubules, as we discussed, they are typically present in the centrosome region, and they are helping in microtubule formation. You can see the first diagram, which are blue colored microtubules which are formed. The second is microfilament or the actin filament, which are present at the boundaries, and they are the double helical structure made up of actin monomer that we'll be able to see in detail in the 20th chapter, that is in muscle contraction as well as movement by utilization of various body parts. And the third kind of filament are intermediate filament, which are green in color and they are scattered in the cell. They are randomly available in the cells. They are basically proteinaceous, and they are doing varieties of tasks. They are giving stiffness, or you can say the structural support or the framework to the cell. Okay, so this is all about cytoskeleton. You can if you like the video, you can like the thumb button, and the second important thing is, if you have any queries, you can put the queries in the question format in the comment box below, and you can also contact on the number that is 990 980 4647, and thank you so much.